Welcome to this presentation on sex determination. We're going to be talking about sex determination, that is, how genetics determines the sex of individuals. So here we have a picture of two lions, and it's pretty obvious which is the male and which is the female. So we can ask the question, how is this determined? Now there are different mechanisms by which sex can be determined in animals, but before we do that, let's just give a little bit of background here. Now, species that have two distinct sexes are called sexually dimorphic. Now, sometimes the difference in phenotype is obvious, like with the lions. We also see that in humans, and also the species shown here on the slide. Now, in peacocks and peahens, it's the male that is the attractive sex, while in species like, say, humans, as well as the praying mantis shown here, it's the female. Now, sometimes it's simply a matter of size. Like crocodiles, the male is much bigger. This sexual dimorphism is necessary for the attraction of a mate in order to propagate the species. But now sometimes sex is determined by chromosomes, and in some species, such as turtles, sex is actually determined by the temperature at which the eggs are incubated. Now for this presentation, we're going to stick to the mechanisms of how chromosomes determine sex in animals. Now before we move on to the specifics of sex determination by genetics and chromosomes, let's talk a little bit about chromosomes in general. Now, all animals have a distinct set of chromosomes, which is specific to that particular species. So, for example, humans have 46 chromosomes, as shown here. Now, chromosomes are described as either autosomes or sex chromosomes. In this case, it is the sex chromosomes that determine the sex or the gender of the individual. Now, when we look at males and females of the same species, we see that they have the same number of autosomes. However, we also see that the sex chromosomes are different between males and females, as well as between species. Now, an example that you are probably familiar with at this point is humans. Now, in this case, as in most other animals, females have two X chromosomes as their sex chromosomes, while males have one X and one Y. Now, it is the inheritance of sex chromosomes which determines the sex of individuals, as I said, in most species. Now, what I just described in the previous slide is an introduction to the XXXY sex determination system. And so here we're going to talk about that in a little more detail. Now, in this case, the males are what we call heterogametic, meaning that since they carry two different or hetero sex chromosomes, they can make gametes that contain only one of these two different chromosomes, the X or the Y. And then they can pass either the X or the Y onto their offspring. Females, on the other hand, are homogametic, meaning that since they only have X chromosomes for sex chromosomes, all of their gametes contain the same sex chromosome, and they can only pass the X onto their offspring. Now, there are two mechanisms that exist in nature that use the XXXY sex determination system. First, there is a system used by humans and all mammals, where the presence or absence of the X chromosome determines the sex of the offspring. Now, in Drosophila, or the fruit fly, it's the dosage of the X chromosome, or the ratio of X chromosomes to autosomes, that determines the sex. Now, looking at this table here, we can see that an X chromosome to autosome ratio of less than 0.5 gives the male phenotype in this species, while greater than or equal to 1 gives a female phenotype, and between 0.5 and 1 gives a male phenotype. Now, similar to the XXXY system is the XXXO system. Here, the females are homogametic, just like in the XXXY system, and the males are heterogametic. However, in this case, the presence of one X chromosome, or no other sex chromosome, so denoted by an O, versus two X chromosomes determines the sex, as depicted in this figure. C. elegans, which is a soil roundworm commonly used as a laboratory model organism, uses this system for sex determination. Now, as you can see in this picture of the C. elegans worms, we can see a male worm and all the associated male reproductive organs, and also a hermaphrodite, the XX hermaphrodite, which has both male and female reproductive organs. Now again, similar to the XXXY sex determination system, we have the system used by birds, butterflies, and some reptiles. This is the ZWZZ system. Now, the most important difference here is that the males are the ones who are homogametic. They have two Z sex chromosomes, while the females are heterogametic. They have a Z and a W sex chromosomes. Now, sex is determined by the inheritance of either the Z or the W sex chromosome from the female. In this case, the ZW again is female, the ZZ are male. So here's an example in birds. We have the Z-carrying sperm, which can fertilize either a W-carrying egg and produce a female, or a Z-carrying egg and produce a male. Now lastly, we have the haplo-diplo system of sex determination. This is a mechanism used by bees and wasps. Now in these species, the males are haploid while the females are diploid. Now what I mean by that is the males are composed of all haploid cells and the females are composed of all diploid cells. Now this is possible because the males arise from unfertilized eggs. Now the females produce these haploid eggs by meiosis, and these then develop into males. But the females arrive from fertilized eggs. So the fusion of a haploid egg, produced by a female, with haploid sperm, produced by mitosis in the male. Now the fusion of this haploid egg and this haploid sperm create a diploid embryo, which then develops into a female. This figure shows a summary of the 4 sex determination systems that we've just covered. Now let's stop and consider a few things here. We know that any deviation from the normal number of chromosomes leads to serious abnormalities in phenotype. So for example, individuals with Down syndrome have 1 extra chromosome, 47 instead of the normal 46. So if we have males and females with different numbers of sex chromosomes, taking humans for example, females have 2 X's but males only have 1. Now how can this be? How do we compensate for these differing dosages in sex chromosomes? Well, there are 3 possible mechanisms for dealing with this issue, each of which makes sure that all individuals have the proper number of active X-linked genes, or genes located on the X chromosome. So the first mechanism, which is used in all mammals, is that 1 X chromosome is activated in each female cell. And this inactive X chromosome can be seen in stained cells from mammals as a condensed structure known as a Barr body. We'll get back to this in a moment. The second mechanism is that the X chromosome in males can function essentially twice as hard as those in females. This is a concept known as hyperactivation, and this is what happens in Drosophila. Now lastly, the X chromosome in females can work only half as hard as that in males. This is the case in those roundworms, those nematodes, such as C. elegans. Now let's look more closely at the inactivation of X chromosomes in female mammals. Now again, this allows for dosage compensation in that now we will only have 1 functional X chromosome in each cell in both males and females. Now this X inactivation is random in each cell, so about half of the cells will inactivate the maternally inherited X chromosome, and the other half will inactivate the paternally inherited one. Now this choice of inactivation is maintained in the daughter cells, but all X chromosomes are reactivated in the gametes so that both X chromosomes are functional in eggs. This random inactivation leads to genetic mosaics because half of the cells have active genes from 1 X chromosome and half of the cells have active genes from the other X chromosome. Now an interesting example of random X inactivation and genetic mosaicism is seen in cat coat color, shown here in this figure. Now, in cats, the coat color genes are found on the X chromosome, so that in female cats, what happens is that half of the cells will express the coat color genes inherited from the mother, half of the cells in this animal will express the coat color genes inherited from the father. So what will result is the random expression of different coat color alleles and therefore a calico pattern. Thank you for watching. I hope you enjoyed this presentation on sex determination.